Good afternoon. I'm George Kunosh, and today I have the distinct privilege to introduce the Wednesday afternoon Director Series speaker, Dr. Michael Karin. And I hate to start with a platitude that the speaker doesn't really need introduction, but in this case I think it's more true than in any other that I've recently attended at these meetings. Dr. Karin is known to many of us through his outstanding work and also through his frequent visits to NIH. I understand that he's a very sought-after speaker on a number of our series. He's a native of Israel, as you know, and after getting his undergraduate degree in biology in Israel, he moved to the United States and got a PhD in molecular biology at UCLA. And after a few brief postdoctoral stints, he moved to the University of California at San Diego in 1987, where he's been ever since, and currently he is the Distinguished Professor of Pharmacology and Pathology at that university and also Cancer Society Research Professor since 1999. As an indication of the very important translational aspects of his research, as it will become obvious from his talk, he was a co-founder of cell signaling, which is today called Celgene, and is still on the advisory board, for which we envy him. We can do that here at the intramural program. Dr. Karin's work really focused on some of the most fundamental issues in biology, how cytokines and hormones, particularly those that are involved in inflammation and stress, influence tissue function through modifying gene expression. And in that area, he went into quite depth by identifying various cis elements, novel transcription factors that interact with them, and most importantly, various kinase, protein kinase cascades that control their activity, most notably the Jun kinase group and the ICAP-B kinase. Now, the relevance of his work is really in the area of an interface between inflammation, metabolic diseases, and cancer. And by saying that, I practically, in these three terms, covered about 80% of human pathology, so you would understand then why Dr. Karin is certainly in the area of molecular biology the #1 mostly highly cited author in the world, and I would imagine that even if he took all of biology, he's probably in the top 5. And that's another thing we like to do in the United States, which is not always very attractive, to try to convert quality into quantity, give numbers. But in looking at his very illustrious CV, I couldn't resist the temptation to count his papers on some of the most prestigious journals, and ended up counting 39 papers in Cell, 36 in Nature, and 22 in Science. And that's certainly, those are intimidating numbers, but certainly indicative of the impact and importance of his work. So without further ado, I'll invite Michael to present his talk. I was asked to let you know that there will be a reception after the talk over in the NIH library, so all of you are invited to attend.
Okay, thank you very much, George, for the kind introduction, and as I discovered, also for being responsible for this invitation to speak to you today. And I decided to change the title a little bit because this is to really appeal. I was trying to appeal to the broader audience possible, and I will also try to address a rather rare medical condition. I'm just joking, which is obesity and aging. So these are actually very common problems nowadays, especially in the US. We started investigating the relationship between inflammation and cancer close to 10 years ago. And we were initially driven by epidemiological studies that led to the estimate that a large chunk of human cancer-related deaths occur in the context of chronic infections and inflammation. And just to summarize some of the findings that we and others in this field have made, I can list actually 4 different types of inflammatory situations that contribute in one way or another to tumor development. But I would like to focus mostly on one issue today and also on one experimental system, and at least in mammals, even though I will talk also about Drosophila, and that's hepatocellular carcinoma or liver cancer. But anyway, from studying different types of cancer, we found, as I said, 4 different types of inflammatory conditions or situations. We have the more common kind of condition is that you will find chronic inflammation caused by persistent infections and or autoimmune disease, which underlies the development of the cancer and precedes tumor development by many years. And one can follow this and then ask how this increases risk of tumor development and so on. Other situation is that we have a low-grade chronic inflammatory response caused by some type of environmental or dietary exposure. For instance, exposure to asbestos, silica, or maybe even the volcanic ash emanating from Iceland will be found eventually to increase cancer risk, and again by causing chronic inflammatory response, in this case in the airways, but there are other type of exposure that can lead to chronic inflammation in other organs. But then we have 2 more interesting situations, and in here the results are a little bit more controversial. We have a tumor-elicited or tumor-associated inflammatory response, even in tumors that are not associated with an underlying inflammatory disease, for instance, breast cancer or prostate cancer. And the question is whether the inflammation represents an attempt by the immune system to control tumor growth, or it's actually something that is taken advantage of by the tumor and then directs it back against us. And unfortunately, I'm not going to discuss this today, but we have worked on this quite extensively, especially, as I said, in the context of prostate and breast cancer. And last but not least, again, I will not discuss today, but we have recently published a paper about the issue of therapy-induced acute inflammation. And I'd just like to mention that cancer therapy, by damaging the tissue, even though it's a cancerous tissue, elicits a wound healing response, which is a type of an inflammatory response. And the question is whether this inflammation that follows the killing of cancer cells, surgical removal of tumors, and so on, actually helps and improves the therapeutic outcome, or actually compromises the therapy, as we have shown in the case of androgen ablation therapy in the case of prostate cancer. But anyway, I just, in a way of introduction, I just like to cover some very early studies that we have done to try to understand the role of inflammation in those tumors where inflammation precedes tumor development. And we first addressed this in a classical inflammation-driven cancer, and that's colitis-associated cancer. And by targeting the IKK beta protein kinase, which is essential for the activation of the NF-kappa B transcription factor, we have shown that NF-kappa B is an instrumental player in connecting inflammation and cancer, but it also acts in a highly cell type specific manner. And we found that in intestinal epithelial cells, NF-kappa B mostly have a survival function, and by increasing the survival of pre-malignant cells, it drives tumor development, but it also has a cell extrinsic effects being activated, in this case, in lamina propria macrophages, where it leads to production of growth factors that then act on the adjacent preneoplastic cells and stimulate their proliferation. We and others have shown that some of these growth factors include TNF-alpha and IL-6, and since I'll get back to IL-6 later on, I'd just like to mention that IL-6 signaling leads to activation of the ROS-MEP kinase pathway, but most importantly, in the context of cancer, it leads to the activation of the STAT3 transcription factor. And we have shown that indeed, in the case of colitis-associated cancer, IL-6 produced by lamina propria macrophages leads to activation of STAT3 in the pre-malignant cells, and STAT3 is one of the key oncogenic transcription factors that drive the development of this cancer. And as I'll show you later on, it's quite instrumental in the development of hepatocellular carcinoma. And work done in many other labs have shown that it's actually a participant in an alarmingly large numbers of solid malignancies. And we believe that strategies that target STAT3 will be quite useful. But from inflammation, I'd like to switch now to a more common condition, which is actually related to inflammation, and that's aging. And as you can see in this rather famous painting of Klimt, the human condition changes quite dramatically from young age to old age, and some of the changes are not only associated with wrinkled skin and whiter hair and so on, but there's also a higher level of baseline inflammatory responses in older individuals, and they are more likely to suffer from chronic inflammation of different sorts. And most importantly, in the context of cancer, it is known that aging is actually one of the major driving force. And if you look at distribution of tumor incidence versus age, if you look at individuals that are 40 years or younger, you hardly find any tumors, and this looks at all tumors. But if you look at solid malignancies, it's even lower. And of course, cancer mortality is extremely low in younger individuals, but then it grows up with age, and there's a linear or even exponential growth of cancer incidence and mortality. And people have tried to develop different theories and different explanations, and I would like to offer you some different views on this later on in the talk, and try to come up with some mechanism that may link aging, inflammation, and cancer. So one of the effects of aging is to increase our body mass, and you can see the aging David is getting thicker. And not only aging increases cancer risk, but obesity increases cancer risk. And when this was plotted against different types of cancer, it was found that the one cancer that takes the cake is liver cancer or hepatocellular carcinoma, especially in men, where increase in body mass index can lead to an up to 4.5-fold increase in the incidence of hepatocellular carcinoma. Some of you may be working on liver cancer risk factors, such as HBV or HCV infection, which makes liver cancer an inflammation-driven cancer, may say that 4.5-fold is not that much, because HCV infection can increase cancer risk by 17 or 20-fold. But you have to realize that in the US, only 1.5% of the population is infected with HCV, and up to 50% of the population is infected with obesity. So the total impact of obesity on the development of hepatocellular carcinoma and many other cancers is actually incredibly high, and it's been estimated that at least 90,000 cancer-related deaths per year in the US could have been prevented if people will keep their BMI under 25, which is not all that easy. So with this, I'd like to switch to the one cancer that really shows all this interconnectiveness between inflammation, aging, and obesity, and that's hepatocellular carcinoma. Hepatocellular carcinoma is the most common form of liver cancer. It's a slow-growing cancer, takes many years to appear, and yet it's a major problem worldwide, and even though it's not the third leading cancer, it's only ranks 5 now in cancer incidence, it's still the third leading cause of cancer-related deaths, and the 5-year survival in hepatocellular carcinoma is less than 5%. So it's a highly lethal cancer. And basically, it's resistant to most conventional chemotherapeutics and radiation therapy. And it's been known for years to be associated with chronic liver inflammation, hepatitis, caused by either infections with hepatitis viruses, HBV or HCV, and in more recent years, the connection between obesity and different obese situations in the liver, non-alcoholic steatohepatitis, or non-alcoholic fatty liver disease, and hepatocellular carcinoma has become rather obvious. So we spent quite a lot of time studying some of the mechanisms that drive the development of these tumors in animals. Mice treated with a carcinogen, diet in nitrosamine, and I just like to summarize these studies before I switch to talk about the role of obesity. So DN is actually a procarcinogen. It undergoes metabolic activation in zone 3 hepatocytes, and it leads to very high incidence of hepatocellular carcinoma, especially in male mice. And we tested whether the activation of NF-kappa B by the IKK beta is involved in the development of this tumor. And we thought that if we will block the activation of NF-kappa B in hepatocytes, we will have similar results to what we saw in colitis-associated cancer. And much to our surprise, when we knocked out IKK beta, we actually stimulated and accelerated tumor development, and the knockout mice exhibited a much higher degree of mortality, more tumors, larger tumors than the control group, the animals with the flox allele that retain IKK beta expression. Later on, we found that this acceleration of tumor development in the liver is not unique to IKK beta, and we in Erwin Wagner's lab have noted that when we knocked out another inflammation-responsive protein kinase in hepatocytes, p38 alpha, we also accelerated tumor development. We asked what's common to IKK beta and p38 alpha, and we thought maybe they control a common pathway or a common set of target genes. And much to our surprise, we found little in common between those two kinases and the pathways that they activate, except that the knockout of either one leads to increased accumulation of reactive oxygen species. And we could relate this increased tumor development to increased ROS accumulation. And the most important thing that what we think that the reactive oxygen species are doing, other than having effect on some molecular targets, is that they actually lead to cell death. And the liver represents a rather unique situation, because cell death in the liver actually triggers cell proliferation. And you can see this very well in the IKK beta knockout mice. And if you look at either control mice or the knockout mice, you look at basic rates of cell death in the liver, looking at apoptosis with a tunnel assay, it's extremely low. You give DN, it undergoes metabolic activation in zone 3 hepatocytes, just around this region of the liver. And some of the cells, because they will accumulate reactive oxygen species, we believe, undergo cell death. And you can see that in the knockout animals, there's a lot more cell death. And despite the increase of cell death, you don't have less cancer, you actually have more cancer, because the more cell death there is, the more cell proliferation you find around the region where cell death occurs, and you can identify the proliferating cells by labeling them with bromodioxyuridine. And when you merge the tunnel-positive cells with the BRDU-positive cells, you find that they are in close proximity to each other. And we believe that in the liver, cell death and cell damage drives cell proliferation, and that's critical for tumor development, because the main difference between the column in where NF-kappa B actually drives tumor development in epithelial cells, and the liver, where NF-kappa B in the epithelial cells actually inhibits tumor development, lies there in their respective tissue kinetics. And whereas the half-life of the average hepatocyte in the mouse is about 6 months, and most hepatocytes, after 2 weeks of age in the mouse, slightly later age in the human, are synchronized in the G0 phase of the cell cycle, and pterocytes in the mucosa are actually replaced every 1 to 2 weeks. They don't live very long, and they're replaced constantly, and damage to the mucosa doesn't really accelerate the rate of cell renewal because the cells are already replaced at a maximal rate, so it's not the bottleneck that determines the amount of cell proliferation in the tissue. But in the liver, a tissue that doesn't proliferate, if you stimulate cell proliferation, that has a major impact on tumor development. So to model all of these results, we have to consider the liver. This is actually a highly simplified representation of the liver. We are looking at only 2 cell types. The major cell type is the parenchymal cell, the hepatocytes. Interspersed between them are resident liver macrophages, or Kupffer cells. We introduce the carcinogen into the mice. In some cells, it leads to induction of oncogenic mutations. But if these cells don't proliferate, they will not transmit the mutant alleles to their progeny. And it doesn't matter how many ROS or MYC genes or whatever you will activate, you're not going to end up with cancer. But in quite a few other cells, exposure to DN leads to cell death through accumulation of reactive oxygen species, for instance. And we postulate that these dead cells somehow communicate with the living hepatocytes, and we know that the communication involves the activation of Kupffer cells, which then produce interleukin-6 that stimulates the proliferation of the residual hepatocytes. And when you have this one wave of proliferation, now you amplify the number of initiated cells, and then additional events can occur, and that's where we don't really know what's going on anymore. And eventually, tumors will develop, and if I'll have time at the very end, I may address this aspect. So another interesting and intriguing feature of hepatocellular carcinoma is that it exhibits a marked gender bias. And as I said initially, obesity increases liver cancer risk in men, but not so much in women, just because women don't develop a lot of hepatocellular carcinoma, so there was not too many cases to look at least in the US. And we have shown that part of these gender differences depend on differential production of IL-6, which again in the DN model is produced early on by activated Kupffer cells, which are actually activated by components released by dying hepatocytes. And we know that one of these components is actually interleukin-1 alpha, which is stored in large amounts in normal hepatocytes, released upon cell death, activates a receptor that then transmits the signal via the MYD88 adapter protein, leading to activation of IKK beta, NF-kappa B, driving the transcription of IL-6. And then in females, where estrogen receptor alpha is activated, you get lower production of IL-6 because this is a repressor of the IL-6 gene, and lower production of IL-6 leads to less compensatory proliferation, and in this case, less cancer. So with this, I'd like now to switch to obesity. And we were interested in modeling the obesity in mice, and then using the mouse model to understand how obesity increases the risk of hepatocellular carcinoma. So we have done 2 things. We have combined the administration of DN to 2-week-old mice with exposure to high-fat diet. What we do is we inject the mice with DN at 2 weeks of age, let them recover for about 2 weeks, and then at 1 month of age, we divide the group into 2, half are placed on high-fat diet, 60% of the calories come from fats, and the rest are maintained on normal chow or low-fat diet. And you can see that consumption of high-fat diet increases the number of tumors, the size of the tumors, And if you don't get 100% induction in the whole group with DN, you increase the incidence to 100% when you put them on high-fat diet. Just to make sure that there was nothing toxic in the diet per se, we conducted a similar experiment with OB mice, leptin-deficient mice, which start their life pretty much at the same size, same body mass as littermate controls that produce leptin, but with time will become obese even on normal diet. And when you inject DN into the OB mice, you will find that 8 to 9 months later they will have developed a lot more tumors than the wild-type controls, even on regular diet. In the second model, we delayed the administration of DN until mice were 3 months of age. And what they did in these 3 months, we put them on either a high-fat diet or low-fat diet. And we found that if you delay the administration of DN, something that has been known for a long time, mice on low-fat diet do not develop any tumors. But if they've been on a high-fat diet for 3 months, now DN alone will induce tumors. And in this case, DN would only induce tumors if mice receive the tumor promoter, the lean mice, and that's phenobarbital. And by comparing the effect of phenobarbital in lean mice and obesity in the obese mice, we found that obesity is as potent or more potent than phenobarbital, a well-identified liver tumor promoter in promoting tumor development. So the question is, what obesity does in the liver to promote the development of tumors. So first we looked at various protein kinases related to metabolism, and originally it was suggested that one way in which obesity can increase cancer risk is by elevating the production of insulin. And the reason that obese individuals make more insulin, including obese mice, is they're actually insulin resistant. So they actually don't respond to this increased insulin. And you can tell that they're insulin resistant because you get less AKT activation, both in the normal liver tissue as well as in the tumors. So the original hypothesis that this extra insulin drives the activation of AKT, which is an oncogenic protein kinase that increases cell proliferation, does not seem to apply in the case of hepatocellular carcinoma. However, another protein kinase or another pathway that is clearly activated by obesity, both in the normal tissue as well as in the tumors, and even more so in the tumors, is the mTOR or TORC1 pathway. And I'll talk about this pathway later on. And you can see that TORC1 has been activated by seeing the increased phosphorylation of S6 kinase, a substrate for TORC1, and increased phosphorylation of ribosomal protein S6, which is the substrate for S6 kinase. And you find this both in the normal liver tissue and in the tumors in the obese mice. But we also looked at activation of protein kinases related to inflammation and stress. And we found that, as we already knew, that obesity leads to activation of Jun kinase, which, unlike P38 and IKK beta, actually drives the development of hepatocellular carcinomas. You find increased JNK activation in the normal tissue, especially in the tumors of mice kept on high-fat diet. There's increased activation of ERK in tumors in mice on high-fat diet. And interestingly, activation of P38 is actually attenuated. And then we found that obesity leads to increased activation or increased phosphorylation of STAT3, which I already mentioned, both in the normal liver tissue and in the tumors of the obese mice. And the source of STAT3 activation is most likely the cytokine IL-6 all over again. Its production is greatly increased by obesity. You can now see increase of IL-6 in the circulation. If you look at the liver, there's a lot more IL-6 mRNA in the liver of mice kept on high-fat diet. And then the cytokine that actually stimulates the transcription of the IL-6 gene, TNF-alpha, is also elevated, but you don't see this in the circulation. You have to look specifically in the liver. And again, both the non-tumor tissue and the tumor tissue of mice kept on high-fat diet show higher amounts of TNF. So we ask, what's the contribution of TNF and IL-6 to tumor development? I'm not going to show you the results from the IL-6 knockouts, but the results from the TNF receptor are very, very clear. When we knock out the major, or we obtain mice that don't have the type 1 TNF receptor, tumor development in lean mice is not affected at all by ablation of TNF-R1, but the response to obesity is totally gone in the absence of TNF-R1, and also obesity-induced production of IL-6 is eliminated in the absence of the TNF-R1 receptor. However, TNF receptor deficient mice still get fat, so it's not a cure for obesity, but most interestingly, the distribution of fat within the animal is different. So these animals which are fat, interestingly, do not have much fat accumulation in the liver, and that's where the cancer happens and cancer development takes place. And this is shown for the IL-6 knockouts, but the same results are obtained in the TNF receptor knockout. So you can look at fat accumulation in the liver by staining it with oil red O, and you find that in the absence of IL-6 or TNF receptor, much less fat accumulate in the liver, although in subcutaneous deposits and other fat deposits, fat accumulation is the same. So why is fat in the liver so important? It's because this condition of hepatosteatosis, fatty liver, eventually results in fat-driven inflammation of the liver, or steatohepatitis, and you can see this by seeing increased accumulation of neutrophils in the fatty liver and increased accumulation of macrophages. And again, ablation of IL-6 or type 1 TNF receptor completely reduces this inflammatory response, and we think that that's a major factor that protects from the development of hepatocellular carcinoma. So just to summarize, there are many risk factors for the development of this disease, including HBV infection, HCV infection, consumption of alcohol, exposure to carbon tetrachloride, hemochromatosis, which is a chronic condition that leads to accumulation of reactive oxygen species in the liver. And if you ask what's common to all these totally different etiologic factors, is you find that all of them lead to chronic liver injury and oxidative stress in the liver. And with time, this chronic injury to the liver, which drives cells proliferation, increases the likelihood that initiated hepatocytes will proliferate, accumulate more mutations, and eventually give rise to hepatocellular carcinoma. Interestingly, in the last couple of years, a role for IL-6 in human hepatocellular carcinoma has been confirmed by retrospective studies. It seems to be a very good predictive factor for rapid progression from viral hepatitis to full-blown hepatocellular carcinoma, and we would like to suggest that drugs that either block the production of IL-6 or IL-1 signaling or even TNF signaling in the liver may reduce the risk of this disease, as well as antioxidants. With this, I'd like now to switch gears and talk about the TOR pathway and its possible role in liver metabolism, but actually in liver cancer, studies that are still ongoing, but in several other diseases as well, including aging and age-related diseases. So we actually started our venture into this by looking at p53, and p53, as you know, is one of the major stress sensors activated by genotoxic stress as well as oxidative stress and other types of genotoxic damage, and is one of the most important, most frequently lost tumor suppressors in human cancer, and it's a major tumor suppressor also in hepatocellular carcinoma. The reason that we became interested in p53 in hepatocellular carcinoma is that it's been shown first by Arnie Levine's lab that p53 can also inhibit the activation of TORC1, which is actually elevated in the obese mice which develop more cancer. And the question is how p53 leads to downregulation of TORC1 activity. So it's been known for quite some time that p53 activation can stimulate apoptosis, and if there's not enough, if there's just a little bit of DNA damage and the cells can repair it, it actually leads to growth arrest and can also trigger senescence. But in the last few years, it was also shown that p53 can inhibit glycolysis, can inhibit protein synthesis, and can actually stimulate autophagy. And we think that these two activities are probably all accomplished through its effect on TORC1. So what is TORC1? TORC1 is a protein kinase complex assembled around the catalytic subunit mTOR. mTOR stands for mammalian target of rapamycin. So this is the protein kinase whose activity is inhibited by the immunosuppressive drug rapamycin. There are two complexes, TORC1 and TORC2, and most of the activities I will talk about involve the TORC1 complex, whose significant feature in addition to mTOR is the presence of this scaffolding protein raptor. And activation of mTOR, of TORC1, leads to stimulation of protein synthesis through activation of S6 kinase, as I mentioned, phosphorylation of the translational inhibitor 4-EBP. It also stimulates ribosome biosynthesis. It has effects on transcription, and I will spend quite some time about the role of inhibited autophagy. And this is the major negative regulator of autophagy. TORC1 is regulated through a rather complex series of inputs. There are inputs from growth factor receptors that go through the exchange factors TSC1, TSC2, tuberous sclerosis complex that are negative regulator of the small GTPase that activates TORC1, RAB. But another very important regulator of TORC1 is the protein kinase AMPK, which is the energy sensor of the cell. AMPK is activated when energy level in the cell drops down, when the AMP to ATP ratio increases, so lower ATP production, higher levels of AMP in the cells activate AMPK, and through phosphorylation of TSC components as well as Raptor, activation of AMPK leads to inhibition of TORC1, and by doing so, it can stimulate autophagy. So we ask how p53 can control this, and this time a new fellow joined my lab, Andrei Budanov, who was involved in the discovery of a new family of proteins, stress-regulated proteins, two of which, cestrin-1 and cestrin-2, are activated by p53. The third one is regulated, cestrin-3 is regulated through the FOXO pathway. When Andre joined the lab, he knew that these have some tumor suppressor activity, they may have antioxidant activity, but he had no clue how they accomplish their activity biochemically. And through a series of studies, he eventually found that what the cestrins do in response to their induction, they are not expressed at very high levels in non-stress cells. They accumulate when p53 is activated, and then they interact with AMPK. They facilitate the activation of AMPK, and by promoting the activation of AMPK, they lead to inhibition of TORC1. So for instance, if you treat cells that express cestrin-1 and cestrin-2 with camptothecin, a genotoxic drug, you see decreased phosphorylation of S6 kinase, decreased phosphorylation of S6. This depends on p53, because in the absence of p53, there's no effect on S6 kinase activity. And in these cells, it depends on cestrin-2. In other cells, it depends on cestrin-1, which are very important for the effects on TORC activity, but are not involved in the induction of p53 target genes such as p21 and other transcriptional outputs for p53. This is quite important in the liver, where we found that cestrin-2 mediates the inhibition of S6 kinase in the liver in response to genotoxic damage induced by DN, our carcinogen of favor. And you can see that DN administration leads to inhibition of TORC1 by looking at phosphorylation of S6. And again, the cells that no longer stain with the phospho-S6 antibody are located in zone 3 in the liver, the site where DN is activated. And this response is highly attenuated in the cestrin-2 knockout mice. However, to fully understand what the cestrins are doing in mice will take some time, because we have 3 genes, 1, 2, and 3. Biochemical studies tell us that we cannot find any differences between the 3 of them. The regulation is different, but the function of the protein products is identical. So to understand what the cestrins are doing, we went to a model organism that has a single gene of this family, and that's Drosophila. Junhee Lee in the lab, who is a very skilled Drosophila geneticist — my lab is not a Drosophila lab — has managed to obtain flies that can either overexpress cestrin or are null for the cestrin gene. Interestingly, when you look at expression of cestrin throughout development of Drosophila, you find that actually as the flies get older, they express more and more cestrin. And then when we look at adults of different ages, which is not shown here, the older the flies get, they express higher and higher levels of cestrin, and the tissue with the highest level of expression is the muscle. So we ask, how does, if cestrin expression is also subject to induction in response to genotoxic stress? And as much as we tried, we could not find any evidence that activation of p53 in Drosophila would lead to cestrin induction. But eventually we found that chronic activation of the TOR pathway in response to regional expression of constitutively activated insulin receptor that leads to AKT and TORC activation results in elevated expression of cestrin. And this was totally dependent on accumulation of reactive oxygen species, which led to activation of Jun kinase, our old friend, and the transcription factor that mediated this effect was FOXO. So this suggested that not only cestrin inhibits TOR, but TOR induces cestrin, and it's likely to be part of a negative feedback loop in which TOR activation would actually lead to eventual inhibition of TOR activation. So to understand the full impact of this loop, we first looked at the biochemical activity of the Drosophila protein. We overexpressed the Drosophila protein in mammalian cells, and we found that it leads to as potent inhibition of S6 kinase and TORC1 activity as the mammalian protein, and under these conditions you get complete inhibition of S6 activity. And then we express both the mouse protein and the Drosophila protein, for instance in the Drosophila eye, and we have shown through its ability to inhibit TORC1, which is the major mediator of cell growth, it can decrease the size of this gigantic eye, which is driven by chronic activation of insulin receptor. And again, the mammalian proteins are as functional in Drosophila as the Drosophila protein. But this doesn't tell us what's the physiological function of the Drosophila protein. So to do that, Junhee obtained cestrin null flies and started characterizing them And in the beginning, it was rather disappointing because these flies developed normally and they look totally normal. But then he did not give up, and he started dissecting the flies and looked at different organs. And because we are interested in the liver, he looked at the fly liver. So flies have a liver. It's just called the fat body. And the fat body has fat, but if you don't have cestrin, it has more fat. And it turns out that the total body content of triglycerides in the fly, which are mostly located in the fat body, but not only in the fat body, is increased by 35% in cestrin-null flies. And he linked this to decreased activation of AMPK. AMPK activity was only decreased by about 45%, but it was highly reproducible. And as a result, there was a 23 or 25% in TORC1 activity. So you say this 25% increase is not that much, but I wish I will get a 25% increase in my salary over a long time. So over a long time, 25% does quite a lot. And we ask whether these small changes, small biochemical changes, were involved in the first phenotype we identified, which is the lipid accumulation. So what we did is we placed wandering larvae on meal supplemented with different drugs. We use a pharmacological approach. We gave them either an activator, a direct activator of AMPK, ACAR, or the anti-diabetic drug metformin, which leads to activation of AMPK by decreasing ATP production by mitochondria. And when you give them ACAR, or especially metformin in this organ, the metformin treatment is as good as re-expression of heat shock inducible cestrin in the cestrin-null fly in inhibiting lipogenesis. You can also inhibit lipogenesis by giving the flies rapamycin, which, as I said, is the direct inhibitor of TORC. So with this, we decided to look for other phenotypes. And because obesity is associated with heart disease, and also heart muscle and skeletal muscle are sites of high cestrin expression in the fly. And most importantly, I met Rolf Bodmer at the Burnham Institute, who is a Drosophila cardiologist. He offered to look at the Drosophila heart. And to many of you, it may come as a surprise, but Drosophila has a heart, and just like our heart, it pulses, and it pulses in a regular manner. And you can see the wild-type heart pulses very regularly, but the cestrin-null heart, you can see, can miss a bit now and then, and it actually exhibits cardiac arrhythmia. And you can correct this cardiac arrhythmia by giving the flies either ACAR or even better, rapamycin. So in this case, rapamycin works extremely well in preventing loss of cardiac function. And with this, we shifted to the major muscle in the fly, and that's the flight muscle. And when we look at 20 days old flies live for about 90 to 100 days, so it's equivalent to a 20-year-old human or a 16-year-old human, assuming we live for 80 years, and they live for 100 days. You find quite remarkable loss of sarcomere structure, complete muscle degeneration, and accumulation of swollen abnormal mitochondria. And despite this almost complete loss of muscle structure, you can restore it either genetically by re-expression of cestrin, which is not surprising because these are cestrin-deficient fly, but you can do this in the cestrin-null flies by giving them, again, pharmacological agents that would lead to the activation of AMPK. You can do it with ACAR. You can do this with metformin. It also works with rapamycin. And more interestingly, it also works with vitamin E, which is an antioxidant. So why does the vitamin E work is that when we look at the muscle, the degenerated muscles in the fly accumulate more reactive oxygen species. And actually, this accumulation of reactive oxygen species precedes the structural deformity of the muscle by about 10 days. So we can already see increased ROS accumulation and decreased mitochondrial function measured by a cis-aconitase assay in the null muscles about 10 days before we see this complete degeneration. And again, what you see accumulating in the null muscle already at 5 days are swollen mitochondria. It really gets real bad around the time you see the complete degeneration at 20 days. There's a lot of cell death in the muscle, apoptotic cell death, as identified by a tunnel assay. And interestingly, you see accumulation of ubiquitinated protein aggregates, the kind of aggregates you see in neurodegenerative disease, Parkinson, and Alzheimer's disease, but here you see them in the muscle. So when we consider all of these signs, it suggested us that maybe the culprit is defective autophagy, which, as I mentioned to you, is regulated by mTOR. mTOR activation would lead to inhibition of autophagy. And why we suspect it's autophagy? Because a variant of autophagy is involved in the clearance of damaged mitochondria. And this is extremely important in cells such as muscle cells and neurons, and also hepatocyte cells that don't proliferate. They somehow have to live with their mitochondria for a very long time. So even though mitochondria can divide, they don't divide all that well. And there are, you know, like your car engine, if you don't take care of it and you race it way too often, it gets damaged. And the way to take care of damaged mitochondria is to engulf them with autophagosomes. And this happens in the absence of starvation. You get rid of the damaged mitochondria. You prevent it from producing reactive oxygen species, something that damaged mitochondria do, and can then damage other mitochondria. You recycle all the components of the mitochondria by degrading them in the lysosome, and then you make new mitochondria. So autophagy is very important for keeping the health of mitochondria within the cell. So to test whether defective autophagy can lead to similar phenotypes, we ask how TORC activation inhibits autophagy. We didn't do the experiments ourselves. We looked at the literature. And it's been proposed that there are two major targets that are phosphorylated by the TORC1 complex. One of them is ATG1, another one is ATG13. And the phosphorylation of ATG1 with a positive regulator of autophagy inhibits its function. So to mimic this chronic activation of ATG1, what we did is we silenced expression of ATG1 by expression of cell-type specific siRNA, either in the heart or in the muscle. And basically, when we did this, we got the same cardiac arrhythmia, even worse than with what we saw in the sestrin null flies. We got muscle degeneration that looked very similar. We got increased accumulation of reactive oxygen species, ubiquitinated protein aggregates, and the whole sequela. So I'd like to propose that defective autophagy is not only important in the fly, it's probably important for all of us. And we think that perhaps it's doing this through increasing the accumulation of reactive oxygen species, also increased accumulation of ubiquitinated protein aggregates. It may contribute to aging, tissue degeneration, obesity, as well as cancer. And there's quite a lot of circumstantial evidence for that. Rapamycin has been shown to increase lifespan in a variety of organisms, including mice. Caloric restriction, which lowers ATP production, and it's the best and cheapest way to stimulate autophagy, increases lifespan and prevents tissue degeneration in many circumstances. And there's also epidemiological studies now showing that metformin decreases cancer risk, And I just heard that metformin was found also to lower the incidence of Alzheimer's disease, which is also associated with accumulation of ubiquitinated protein aggregates, which may not actually be the cause of the disease. They could be just a symptom for the disease. And actually what goes wrong in a lot of this degenerative disease, it could be the process of autophagy. And that's, I think, an hypothesis that merits further investigation. Finally, I'd just like to go back to cestrin in the mice. If it controls metabolism in the fly, does it control metabolism in the mice? So far, the only knockouts we have is of cestrin-2. The liver expresses quite a lot of cestrin-2, even before stress. So what we did is we placed mice, either wild type or cestrinal mice, on high-fat diet and performed glucose tolerance and insulin tolerance tests. And you can see that the absence of cestrin-2 results in systemic insulin resistance. And then when we use clamp studies to determine the site of action, we find that it only affects insulin signaling in the liver and decreases sensitivity of hepatocytes to insulin signaling. And this is associated with decreased activation of AKT. In more recent experiments, we actually crossed these mice to the OB mice, which are insulin resistant but are not diabetic. And it looks like the absence of cestrin-2 causes frank diabetes. Glucose is elevated up to 600 milligrams per deciliter. So I'd just like now to end and acknowledge the people who did first the second half of the work, the Drosophila work and the cestrin work in the mammals, which a project that was initiated by Andrei Budanov, a very talented postdoctoral fellow who is actually looking for a position. So if anybody has anything, Andrei is out there. Junie Lee joined the lab more recently, is an expert on Drosophila, and in collaboration with Ethan Beer's lab and Rolf Bodever's lab, which are Drosophila labs, we were able to do all of that. Mark Ellisman's lab helped us with the EM analysis, and Bill McGuinness' lab, another Drosophila lab, also helped us with some of the Drosophila studies. And finally, I'd just like to acknowledge Shin Maeda for starting the HCC project in the lab. I mentioned some work on ROS accumulation in the liver that was done by Hide Kamada. Toshi Sakurai did the P38 knockout. They're all back in Japan, running their own labs now. And Scott Nogler did the IL-6 work. He's now in Oregon. And Grace Yu, Gubin He, and Ik-Jung Park did the work on obesity and STAT3 that I presented to you. And thank you for your attention.
So I was wondering, with the IKK knockouts that get the increased tumors, did you notice an increase in the infiltration or proliferation of immune cells in those tumors? And also, are they fatter than the wild types?
Yeah, I mean, we looked. It's a little bit hard to quantitate because the tumors in the knockout are so much larger. So the answer is we have not done this thoroughly, so I cannot answer the question. But you really have to do very careful analysis and measure the infiltrating cells per surface area, per volume, and so on.
Michael, in your cartoon, you depict cestrin acting on AMPK. Do you have any idea how it works, and is this consistent with data on LKB1?
Okay, the regulation of cestrin by junk?
No, the regulation of
of AMPK. Okay, yeah. So the dependency on LKB1 is not fully clear. When we overexpress cestrin in LKB1 knockout fibroblasts that we got from Ruben Shaw, it still activates AMPK. So under these conditions, it's LKB1 independent. But when we express it in HeLa cells that also don't express much LKB1, it doesn't activate AMPK all that well unless we supplement it with LKB1. So it can be both LKB1 dependent and independent. It probably works through a similar mechanism and probably increases phosphorylation by LKB1 or by some other LKB1 like kinase that is present in the LKB1 knockout, and it depends on how much cestrin we succeed in expressing in the cells. And even in Drosophila, we try to do this with LKB1 knockout flies, and the results are not all that clear because I think we need to titrate cestrin expression very carefully and measure the LKB1 dependence vis-a-vis the level of cestrin expression.
Thank you.
Yeah.
Is it on? I have two questions. One is with regard to the TNFR knockout when it comes to see the obesity-mediated tumor progression. Is this solely attributed to the TNFR1, or you get any data from TNFR2 knockout? And the second, when it comes to the high-fat diet, do you have any data regarding the fate of glycolysis? I mean, does the cell chooses to utilize it for ATP production, or as a tumor progression, it utilizes the pathway to get intermediate metabolites?
Yeah, no, these are very good questions. So first, the easy one. We look at TNFR1 because we presume it's acting within the hepatocyte because these are the cells that accumulate the lipids in the hepatosteatotic liver. It's not infiltrating adipocytes. And we knew from many studies that we and others have done that the major TNF receptor in the hepatocytes is TNFR1. So we only looked at TNFR1 knockouts. So I cannot tell you if TNFR2, which is not expressed at very high level in hepatocytes, can contribute to this at all.
I have no idea too. That's why I was asking, just in case there's something.
So I have no idea. The answer to the other question, we haven't looked at that. You really have to, to measure all aspects of metabolism very, very carefully, and I just don't know. All I can say is that, the inhibition of glycolysis by p53 has really been demonstrated in cultured cells. Yeah. And even though there are p53 knockout mice, there's not much evidence that p53 will regulate glycolysis in the entire metabolism. And I think if it was a major regulator of glycolysis, and you would have runaway glycolysis in the absence of p53, if you knock out p53 in the liver, you should have major metabolic derangements, and you don't have them. So you can knock out p53 in the liver and the mice are fine. They're more susceptible to tumor development. But I think we need to look at metabolically what's the high-fat diet doing and so on.
It's a very good talk. I learned a lot. I want just to ask you, there have also been studies where the menopause and obesity are also related to cancer progression or development. And your knockout females, you assume they are protected from the transformation because of the estrogen. So I was just thinking if you are planning in doing some experiments in that regard, in removing ovaries from your young female knockout, feed with high fat, and see in females now, for one time, we know about males. I would like to know what we can learn from, for females from your model.
We need to do these experiments with the obesity model. We have done it with the regular DN. So the female loses the protection when you remove the ovaries. They become much more susceptible to induction of HCC. And when you look at women, the ones who get HCC get it at a much later age than men. It's usually in mid-50s. So it correlates also with menopause in women. And you can protect the overrectomized females by giving them estrogen or ER-alpha, a pure ER-alpha agonist. But we have not looked in the context of obesity. If we put females on high-fat diet, we can stimulate HCC, but I did not go through that, and it's very similar to what you find in humans. They get more cancer, but it's not a whole lot more. It's not like in the male. The obesity in the male, the effect is much stronger than in the female, even though the female starts with a lower baseline.
Michael, thank you for a nice talk. I have two questions. First question is, you told us cystrin knockout, Drosophila looks to have autophagy deficiency. Did you check some of experiment with autophagy assay? It's my first question. My second question is, did you observe some of the difference in cystrin wild type or knockout Drosophila in calorie restriction condition?
Yeah, good question. So in Drosophila, it's very difficult to identify autophagic vesicles. So we tried. We did not succeed. So the evidence for the autophagy is mostly genetic and based on all these other signs and so on. And by the way, if you knock out ATG7 in mouse muscle, you get exactly the same phenotype that I just described to you for Drosophila. I mean, every single aspect. There was a paper published by an Italian group in Cell Metabolism. In the mouse liver, we are now looking at effects of cystrin to knockout on autophagy, and there it's much easier. So we do see effects on, LC3, 1, 2 processing, P62, and so on. So it does have effect on autophagy. What's more complicated is the starvation experiments, because when you do acute starvation, you actually stimulate lipolysis in adipose tissue, and the liver gets actually filled up with lipids. And you actually see this in mice that cannot mount enough autophagy, that even under starvation, when you're supposed to stimulate autophagy, if they have defects in the liver, you actually get, even though you starve the mice, you see now more hepatosteatosis. But we are doing all of these experiments.
So several years ago, Song and I suggested that there are damage-associated molecular patterns that would turn on the innate immune system, and that one category of those would be exposed hydrophobic portions. So from that, we would predict that your cystrin knockout mice, which are hyperlipidemic and so on, would have either higher constitutive levels of immune antimicrobial products, or would be more sensitive to stimulation of them. Have you noticed either of those?
Yeah, I mean, they showed me the people in the lab showed me some effects on T cells that they were pretty striking. I don't remember what they were. It was something to do probably with Tregs or Th17 or I just can't recall, because I just couldn't understand it. And I told them that we need to focus on the insulin resistance and the liver.
Send me those results and I'll help you understand them.
Yeah, I think we need to look more what happens in the T cells and the immune cells, but we can only do a few things at a time.